exome sequencing, where it will sequence all the protein coding parts of your gene. And then we now have single-cell genomics now. Now, with this one, it focuses on the genome of individual cells. You know that our cells also have different genomes. And sometimes there's something called mosaicism, where there'll be partial inheritance. You know, there's some funny thing in one particular cell, it may be different from the next cell. Okay, I can use this one. So, single-cell genomics is now even more specific. So there's a lot to get into. And I keep telling people that we can sit and complain that we don't have money and all that. But fine, let's start from the basics. The basics are so important. And I'm glad I'm talking to Faculty of Science. You understand? We are so relevant in this field. It's so, medicine without science is not going anywhere in these current times. That's the truth. There's a lot to do together. The doctor in the clinic will get frustrated if all he's doing is prescribing drugs that are not working. My own PhD started with a doctor like that. They've been seeing children with severe malaria in the clinic. And he said, why is it that one child will develop cerebral malaria in a family and the others will not? What is the cause? And there are many reasons why. There's the child's genetic makeup. There's the immune response. You understand? There's even the parasites virulence as well. So there are so many things we need that we can do, that we can start from the basics. You know, each time I'm talking about these highfalutin tools, I keep coming back to the basics. Because when we look at the highfalutin tools, we become discouraged. But that's why I keep coming back to the basic ones, because we need that basic. It's like building a castle. Without a foundation, there's nothing. So I'll keep going on. So genomics is so important for infectious diseases, for pathogen diversification, to know the different infectious diseases. We can track infectious diseases in our population. We can track the changes. We can track when we have large, like we had COVID, but we don't even, we can have different outbreaks. And we can type the last outbreak with the current outbreak. What has changed? What is different? How can we prepare for the next one? If it mutates, we can predict that it may mutate this way. So we'll be expecting the next outbreak. So there are many things to do. We can work with HIV, tuberculosis. There's a lot of work that has been done on malaria. See the huge amount of details we have from COVID-19 just because of these tools that we have. So these are things, there are even different projects now, various platforms that support pathogen identification, outbreak tracing. There's something called ID-Seq, Next Strain, for those that work particularly in infectious diseases. There's a lot of tools available. There are many projects around that we can join ourselves to, that we can write and, present our data. Our data is so important, even in the global space. So those are things we can do. There's an example of a center, the ASG, the African Center of Excellence. Started from somewhere. Today, they played a key role in the Ebola, even in the COVID-19. And even with a lot of collaboration, they were able to have rapid diagnostic tests that were developed. Even in NIMA, they developed a rapid diagnostic kit for COVID. So there are so many things we can do. We can work with people that have experience, we can learn from them, and we can do the little that we have. Our data is so important. Even we can get variations from different, many of these networks have typed these variations, and they are looking for people who will come and do analysis on them. For example, malaria gen has a lot of variants on P. falciparum. And we can use this data we can start thinking about how to develop vaccines. Do you know that during the COVID-19, it became so very obvious that we don't have any vaccine-producing centers in Africa, even till now. Even till now. So we need to get into this discussion, get into this conversation. I'm just going to run through these cancer treatment options. There's a lot that has happened or is happening right now with genomics. For example, I'm giving an example. This particular cancer, non-small cell lung cancer, they identified what they called an epidermal growth factor receptor. That there are two important biomarkers that they found that were associated with this cancer. And then they discovered that these receptors, they bind ligands like EGF and cause a lot of dimerization and activate pathways for cellular growth. And they found another one, that is the anaplastic lymphoma kinase. These are two important markers in this lung cancer. Now, they found that the EGFR has some particular mutations and discovered that these mutations can be targeted by an inhibitor. So it is that inhibitor that they now took out and, molecular targeted therapy. That's what they call it. So it's like finding an active ingredient in a plant. You will take out the active ingredient, you will process it so that you can use it to manage the disease. So this inhibitor is from humans. So they now found a way to synthesize it. And they are now using it as targeted molecular therapy for this particular cancer. Now, can I shock you with something? That even though this works, over there, it may not work here. You understand? Even though it's worked, they've developed it, they are using it in the US, it may not work for us in Nigeria. For example, for prostate cancer, these are a list of genes that have been associated, that they've typified and found that, you can use it either for PSA screening, prostate cancer screening, and, rapid diagnostic kits and things like that. In Nigeria here, when my student presented this as his MPhil, the internal examiner was so excited because they found out that the PSA levels that they are now using is not, they are not finding it very useful in diagnosing prostate cancer in Nigeria. And so he now said, if we can find genes that are associated in our own population, is it possible for us to develop our own kits so that it's easier to quickly, identify if a man has prostate cancer or not? Because they found out that the PSA levels were not working anymore for our own population. So you can see that there's a lot that we need to do, a lot we can do. There are many things that are going on. They even use, from these data that they have, they can use it to predict risk scores. After predicting risk scores, they will now see a particular individual that if they put all this together, all this is genes, is phenotype and everything, they now put it together. It's like an algorithm trying to find out that, okay, can we predict this person having this cancer, and how long will it take for it to become severe? So there's a lot that is going on. And I want to just challenge us this afternoon. I don't want to go into all this, we're in the afternoon. I don't want to go into too much technical details because we all get bored. That's why I keep interjecting the challenges I am throwing to us. So, for example, this person, there was this polygenic risk scores for coronary artery disease. They used what they call, it's a genome-wide polygenic score to identify individuals that had a higher risk of this coronary heart disease. And so they look, they have a lot of data. That's why I said, let's get young people also involved in bioinformatics. You understand? So how can we use all the results we have from different studies? You know, Nigerians have contributed to many GWAS studies. How do we pull all those GWAS studies, you understand, together from different sources, and then look at our own population and be able to type what are the polygenic risk scores with all this data that we have from our own population in different studies. And from this person's study, they were able to discover that there were some individuals that were at greater risk, you understand, using this particular algorithm. So there's a lot for us to do, whether it's basic laboratory science, whether it's wet lab, as we call it, or dry lab, which is bioinformatics. There's a lot to still do. That is my challenge this afternoon. I don't know if we've all heard about what we call CRISPR-Cas9, clustered regularly interspaced short palindromic repeats. In fact, that's a long thing, so that's why they call it CRISPR-Cas9. So it's used for genome editing. In simple language, it cuts the DNA. And then, normally DNA has what you call repair enzymes. So those repair enzymes will now repair the DNA, but they would have interjected the repair, they've cut the bad, let's put it that way, the bad SNPs, and interjected the correct one in, and then there's an enzyme that will now repair the DNA. That's CRISPR-Cas9 in simple language. And it's been used for cancer, and it's being used for sickle cell anemia now. So, and you know sickle cell anemia is one of the most common monogenic diseases in humans, and Nigeria has a large burden of sickle cell. I want to ask a question here. Are people really aware of sickle cell disease? Because to me, I think the awareness has reduced now. And some people, we are seeing young people making that mistake again of getting married to, you understand, and saying love is all that matters. So my question is, are we really doing that awareness? Because it is critical. Anyway, what I'm doing here is different. So there's a mutation in the gene, we all know. And we have found that children with fetal hemoglobin levels, even towards adulthood, have less severity. So how do we enable development of cost-effective genetic effects? So what they did with this is that there's a cell-based gene therapy, and they will take the patient's stem cells, modify it by genome editing, and then transplant it back into the patient, and then increase the production of hemoglobin F. So with that, it reduces the sickling, and then this person can live a normal life. But again, this kind of therapy is very expensive. At least for now, it is still very expensive. So there's still research that we can still continue to do to make a difference. So what are the challenges for implementing genomics in healthcare? We just mentioned the first one: cost, accessibility. Data management is the second one. And that is why I have been saying that repeatedly. We need people who will develop interest in data management, managing vast amounts of genomic data. We need that expertise in our own population. And how do we now integrate this genomic data into our electronic health records? So there has to be a talk between the basic and the clinical. We cannot work in different places if we want to talk about healthcare in our population. So we need to find a way where we will work together. We generate the genomic data, we interpret the genomic data to the clinician. You understand? So we need to understand how we are going to work together doing this. And then there are other ethical issues. We have to talk about religion too as well, legal and then social issues, cultural issues as well. If these things are presented to people, how would they take it? Would they accept it? So those are the issues we have to talk about in implementing genomics in healthcare. informed consent. I call it understood consent. How many times do people really understand what we are saying? So we need to do a lot of that, informed consent, so that people understand what they are doing. And I believe that the layman is intelligent. If he understands what you are talking about and he knows the future impact that can be even to his own family, so we need to work with that, informed consent. And then if we have all this evidence, or if we do not have the evidence, so there are two ways. If we have the evidence and we don't have the connection with the clinic, how is it going to work? If we do not have the evidence, how will the clinician be able to help us? So those are the two major things in terms of challenges of genomics. So, one, we have to get the data. Two, we have to work together. So those are the major things that I see as issues. So future direction, we need to have a comprehensive training for all of us, putting together basic genetics, genomics, molecular biology, and clinical applications. Like I said before, we cannot build a castle without building a foundation. We need the basics before we can move anywhere. Then we need to start thinking about genetic counseling. This is so important. And as a medical school here, it's something we need to start thinking about, genetic counseling. And genetic counseling is not just the clinician, it involves the geneticist, it involves everybody, in every sphere of life. Those are the important things we have to think about. Then we need to emphasize the importance of ethical, legal, and social issues that relate to genomic medicine. We need to understand data interpretation skills, encourage health informatics. Again, I keep saying it. We need health informatics, we need information technology training, we need bioinformatics. These are the things we need. And we need to start introducing genomic medicine in every curriculum. Let's become aware. The biophysicist has a role to play. The chemists have a role to play. So how do we infuse all these little things in our curriculum? No, even if it's just a course in the whole year, in the 4 years, at least the student has an understanding that I can also play a role in genomics and healthcare. This is my recommendation of future direction. So genomics is so important. The insights will revolutionize our healthcare. And we need to be awake in Nigeria. We need to collaborate with one another. Science, we cannot stay in one place. We need to move together with the clinicians. We need to be able to talk to each other, because at the end of the day, it's our collective responsibility as a population. Thank you very much for listening to my presentation.
Thank you, ma'am, for that lucidly delivered lecture. Before we move on, do we have one or two reactions from the house? Maybe there are things you want to clarify. Okay. One of the graduate students come here now.
Good afternoon. Thank you, Prof, for that lecture. I just want to comment on the area of data management and bioinformatics. But somebody like me in computer science and statistics, we are so much interested in data mining. And then if we look at what artificial intelligence has provided for us today, it has given us opportunity to bridge the gap and the challenges we are facing formally in our attempt to analyze some of these genetic diseases. But looking at our university and some of the things you said on the fact that we are limited in terms of data that domicile in our population that we can use to model genetic diseases, that is completely missing in our own population. But we have several of them online for other population. Then my question is, what is our own Center for Genomics and Molecular Biology is doing to make some of these data available so that we can apply some of these artificial intelligence that are available today to address that area? That's number one. Number two, is there anything that the center can do in order to enrich our skill so that we can actually prepare ourselves and then acquire more skill in the analysis of these genomics data when we look at the AI techniques and those bioinformatics aspects of computational biology? Thank you, ma'am.
Thank you very much for your question. Okay, so for the center, there's a lot that we are trying to do. First of all, we are trying to. It's a long stretch, but one of the ideas we have is to have what we call a biobank. So if we have a biobank, we can collect samples. And the main aim of that is to be able to collect samples, clinical data also collected. And if we have this in our bank, more or less like a bank, somebody has an idea tomorrow and wants to work, he doesn't need to go back to collect that data because data is already available. And all he needs to do is maybe he has a grant, pay a little fee, collect the samples that he wants to work with, and generate data on it. So those are one of the things that we are thinking about, that we could, as something that is a long-term thing, and eventually everybody benefits from it. Now, for data that is already available, there are a lot of data from Nigerian population that are available on genome-wide association studies. I know from my own studies, I have contributed data samples, and that data is available. You understand? So that's what I was mentioning when I was talking, that there are many groups like that have contributed data in diabetes, hypertension, genome-wide association studies. If we have people who are interested in bioinformatics, they can retrieve those data from the different places where they are and pull it together. Let's have a picture of what we have in Nigeria to start with. Now, I'm glad you are interested in something like that. Again, one of the things that we are putting down as a working strategy for the center is to have short-term courses. So short-term courses where you can, at your own pace, you can take the lecture so that you have a better idea about genomics, all these things all I just ran through within 30 minutes. I saw some people were already yawning. That's why I was going fast, so that I don't say anything too technical. But at your own pace, you can learn about these different things, have an idea. So now, even though you are coming from computer science, you have an idea of what you are looking for when you are now using all data mining and doing all these other things. So that is one of the things we're also providing at the center. So we are trying to do that, put that together, and so that we can have people do the courses at your own time. If you want to take a diploma out of it, that will also be available.
Thank you, ma'am. One more. If there is none, I think we should proceed. Now it's time for lunch. I know many of us are saying, thank God, it's time for lunch. But before we go for lunch, please let's listen to this special announcement. If you are presenting if you're having oral presentation today, just go through the book of abstract. You will see 24th. See whether your presentation has been scheduled for 24th. So while we go out for launching, you will see Dr. Adibayo. You can submit your presentation to him. And for those of us that have a poster, during launch, please just go to the back of this hall. You will see we have already allocated what is it called? Number to your poster. Just identify your number and fix your poster there. So the number is already in the book of abstract. Check the title of your poster and go there and fix it to the corresponding number. So this is the schedule of our presentation. Food science will receive a large number of entry from food science. So food science will be in a I think it will, please let's clap for food science. I think food science is taking over Faculty of Science now. Okay. So food science will be having their presentation, oral presentation, lecture room 2. We have 4 lectures room here after here. So lecture room 2 is just, I think the one just opposite, you will see it there. Lecture room 2, we have a technical unit that will be with you to set up the presentation. But that will be after our lunch. So after lunch, just go straight to